ligating the lambda-COS region to the plasmid DNA. The modified plasmid can then be packaged into lambda baryons in vitro. As described previously in the phage particles used to transduce Escherichia coli, plasmid construction avoids necessary of having a transform E. coli, which at best is an inefficient process. One major advantage of plasmids is that they can be used to clone large fragments of DNA. Therefore, fewer clones are needed to obtain representation of the whole genetic element. Plasmids also permit storage of the DNA and phage particles instead of in plasmids. Phage particles are much more stable than plasmids, so the recombinant DNA can be kept for long periods of time. Amplifying DNA, the polymerase chain reaction. Conventional molecular cloning methods can be considered in viable DNA amplifying tools. However, the development of synthetic DNA has spawned a new method for the rapid amplification of DNA in vitro, the polymerase chain reaction. The polymerase chain reaction can multiply DNA molecules by up to a billion-fold in the test tube, yielding large amounts of specific genes for cloning sequences or mutagenesis purposes. PCR makes use of the enzyme DNA polymerase, which copies DNA molecules. The PCR technique requires that the nucleotide sequence of the portion of the desired gene be known. This is necessary because short oligonucleotide primers complementary to sequences in the gene or genes of interest must be available for PCR to work. The steps in PCR amplification of DNA are as follows. 1. Two oligonucleotide primers flanking the target DNA are made on the oligonucleotide synthesizer and added and great access to heated natured target DNA. As the mixture cools, the excess primers relative to the target DNA ensures that most target strands anneal to a primer and not to each other. DNA polymerase then extends to primers using a target strands as a template. 4. After an appropriate incubation period, the mixture is heated again to separate the strands. The mixture is then cooled to allow the primers to hybridize with complementary regions of newly synthesized DNA, and the whole process is repeated. Thus, each PCR cycle involves following 1. Heat denaturization of double-stranded target DNA, 2. Cooling to allow annealing of specific primers to target DNA, and 3. Primer extension to the action of DNA polymerase. How extension products of one primer can serve as a template for the other primer in the next cycle. The beauty of the PCR technique is that each cycle literally doubles the content of the original target DNA. In practice, 20 to 30 cycles are usually run, yielding at 10 to the power 6 to 10 to the power 9-fold increase in the target sequence. PCR at high temperature. The original PCR technique employed Escherichia coli DNA polymerase, but because of the high temperatures needed to denature the double-stranded copies of DNA being made, the polymerase itself also was denatured and had to be replenished every cycle. This tended to limit the number of cycles that could be run and was very expensive. This problem was solved by employing a thermal stable DNA polymerase isolated from the thermophilic bacterium. Thermus aquatics DNA polymerase, T. aquatics known as TEC polymerase, is stable to 95 degrees Celsius. Thus is unaffected by the denaturization step employed and the PCR reaction. The use of TOC DNA polymerase also increased the specificity of the PCR reaction. Because the DNA is copied 70 degrees Celsius rather than 37 degrees Celsius at high temperatures, nonspecific hybridization of primers to non-target DNA rarely occurs, thus making the product of TAC-PCR more homogeneous than that obtained using the E. coli enzyme. One problem with TAC polymerase is that it has not proofreading function, and consequently makes more mistakes than the Escherichia coli enzyme DNA polymerase from the hyperthermophilic archaean Pyrococcus pyrrhus, growth temperature optimum 100%, called PTU polymerase or event polymerase, is also widely used and is even more thermally stable than TAC polymerase. PTU polymerase has proofreading activity, making it a particularly good enzyme when accuracy is crucial. Because a number of highly repetitive steps are involved in the PCR technique, matches have been developed that can be reprogrammed to run through heating and cooling cycles automatically. Because each cycle requires only about 5 minutes, the automated procedure allows for large amplifications in only a few hours. By contrast, such amplification in vivo cloning methods would take several days. To supply the demand for thermostable DNA polymerase in the growing PCR and DNA sequencing markets, the genes for these enzymes have been cloned into E. coli and produced in large quantities. The cost of the PCR method is now just a fraction of what it was when the technique was first introduced. PCR. Applications of PCR. PCR is a powerful tool because it is very simple to perform, extremely specific and extremely efficient. Remember that during each round of amplification, amount of product doubles, leading to an exponential increase. This means not only that one can get large amounts of amplified DNA in a few hours, but that only a few molecules of target DNA need to be present in the sample to start the reaction. The reaction is so specific that with primers of 50 nucleotides and high annealing temperatures, there will be extremely low levels of false priming, and therefore the product form should be relatively homogeneous. Although automated thermal cycling machines are still rather expensive, the technique has found a wide range of applications. Because what can be done with PCR, an analyst of the amplified product can sometimes accomplish in a time few hours what would have previously taken an entire laboratory several days or even weeks to do. Therefore, PCR can be extremely cost efficient and is now used in very wide numbers of practical applications. PCR is extremely valuable for obtaining DNA for cloning and sequencing because the gene or genes of interest can easily be amplified and flanking sequences are known. Because the primers used do not have to be perfectly complementary, PCR is routinely used in comparative or evolutionary studies to amplify genes from a variety of sources where the DNA has already been cloned and sequenced from one organism. In these cases, the primers are made to regions of the gene thought to be conserved throughout the wide variety of organisms. Because of the sensitivity of PCR, it has been used to amplify and clone DNA from sources such as mummified human remains and even samples of extinct plants and animals. PCR can also be used to amplify very small quantities of DNA present in a sample. Using appropriate primers, one can find and identify. Using appropriate primers, one can find and identify a single bacterial cell in a sample even if large numbers of other species are present. Please note that when amplifying DNA from a bacterium, it is not necessary that the organism be grown in a laboratory.